Good afternoon, everyone. A pleasure to talk to you today about some of the research we do with the support of MyCirc at the University of Minnesota. I'm a professor in the biochemistry department, and first, some disclosure: I have co-founded a company that develops some technology that is related to some parts of the talk today. So this is the statement that you see here. What do? We focus on biofouling because biofouling is one very important vector for the spread of aquatic invasive species. Plants, but also seaside organisms can stick to different structures, such as boat, but can also be docks that can be transported from bodies of waters to new bodies of waters, and that can be a tremendous way to spread those species to new bodies of water. This is very well documented across the globe, but this is obviously something that concerns us in Minnesota, and this is one of the reasons why there are some drastic measures into cleaning, scraping, and drying boats and structures when they need to be transported to new bodies of water. And so the current ways of mitigating biofouling, really, to simplify, there are some mechanical ways to do it, and that involves cleaning, scrapping, and what I mentioned now. And then there are some products that we can put on those structures that are called anti-fouling coatings. And those anti-fouling coatings also, in a simplistic way, you basically have two classes of those for now on the market. You have the first class that is non-biocide based, and typically those are silicone-based coating, analogous to the non-sticky pans, if you wish, where reduce the force of adhesion of those organisms onto the surface, and with the hope of reducing that. This works really well for fast vessels, fast ships, and ships that are not immobile in the water, that are not stationary in the port. However, it doesn't work well whenever some vessel or structure is being immobilized. And then the second family, which is the dominant family on the market, is the biocide-based. So these coatings contain biocides, that is, molecules that are toxic. And this is by design because these toxic molecules are supposed to actually kill and harm, the bacteria that are responsible for making that initial slime onto which those larger organisms can adhere. And so really you have those two main classes of solutions, and there is obviously a great need for eco-friendly, potent alternatives to mitigate biofouling and improve our ability to control the spread of aquatic invasive species. This is particularly true because these biocide-based compounds are designed to leak their biocide out. And so you have some marinas, for example, that are heavily contaminated with copper because these, to be effective, these biocides have to constantly release some amounts of these metals into the waters. So they are not environmentally friendly at all, and therefore some alternative is needed. What can we do? Well, what we chose to do is to try to study how this process works, and particularly in sequence. So this is, some hypothesis that we have in the field as to how are things going when you submerge the structure or a material in the water. And some of the first steps that are not on this picture involve, organic molecules adhesion to the surface, but very quickly after, the first bacteria starts to attach to the surface. And these bacteria grow onto the surface, and they form what we call a biofilm. So that is a bacterial structure that includes different types of sticky material onto which larger macroorganisms such as algae, plants, and mussels can more easily adhere. We know that some interesting biological mechanisms are involved in this transition between adhesion and biofilm formation and maturation here, and we'll talk a little bit about that. One mechanism that has been studied in the last 3 decades is called quorum sensing, and it's in fact a type of microbial language. And microbes are constantly exchanging information via chemicals, and those chemicals, those language are fairly analogous to ours. Really, you could see those different molecules here as the different letters of a language, if you wish. And different bacteria could use those different letters, and different combinations of those letters form words and form sentences, and probably convey fairly complex message. And we know that bacteria use this communication system for several key behaviors, including the formation of biofilm. So from there, you know, we thought, well, one interesting approach could be to find a way to blur this communication. What if we can disrupt it? If we can disrupt it, then we should be able to control these bacteria, or at least prevent them from doing whatever they want to do when they communicate. And to disrupt that, we can simply find a way to chemically break down those molecules. So the goal is not to kill the bacteria. So it's a completely different approach from the biocide. We're not trying to kill the bacteria that are involved. We're only going to degrade these signaling molecules here. And the hypothesis is that in normal condition, you have attachment of the bacteria onto the surface, they proliferate, they form this biofilm because they communicate, you have all these signaling molecules, and then you have algae and mussels that are being recruited to that site. Now, the idea is that if we have a molecule that can break down those signaling molecules, we would have adhesion of bacteria, but less biofilm, and consequently, we should have less of the algae, the aquatic plants, and the mussels that are being recruited. So this is the hypothesis. Now, how do we execute this idea? Well, we and many other groups identified enzymes that are called lactonases because they can break this signaling molecule that bacteria use that is a lactone. The enzyme breaks this structure here, opens that ring up, and you have different enzymes here. Those are different representative of different enzyme family. So those are proteins, right? Protein molecules, 100% biological, right? They're just amino acids. And they can break down these molecules naturally. So those are naturally made by bacteria. So we could potentially use them to control bacteria. The issue, though, that we have is that these enzymes, they evolve to operate in the context of cells, to do whatever they have to do in the natural context. Now, if we want them to specifically work against biofouling to control adhesion of mussels, for example, that will require a very different context than a cell, maybe immobilize them into a coating. That's a whole different story. So to do this, we have to select some candidates that have properties that may be compatible with those requirements, like being stable in a coating is very different than being stable in a cell. And one good starting point that one starting point is precisely to identify what is a good starting point, what would be a good candidate for that venture. And those are some of the enzymes that we initially identified and characterized, and we picked two because they have, great stability properties and other aspects that were really appealing, and we further worked with those. Now, characterizing these enzymes, we realized that they have really interesting properties that are induced by their very high stability. That's why we selected them. But they have very interesting properties in the sense that they are fairly durable. So here you can see this is the enzyme activity, so its ability to degrade those signaling molecules over time. This is over 300 days in liquid conditions, so you can see that the activity remains fairly stable over the course of nearly a year, about 10 months. And they can also resist fairly well quite a variety of different solvents, right? So you see that the starting activity here, everything is normalized. So 100% is the activity in water. And you can see that in all of these different solvents, the activity is about 100%. So no significant damage there. Here it's higher in this solvent. And for some of them, like xylene, it's lower. But mostly those enzymes are fairly solvent resistant, which is a good news if we want to include them into materials such as coating. So very desirable trait. And in fact, this is a project that we initiated quite a few years ago now with the collaboration of Randall Hicks in Duluth. This is mostly the work of Sichuan Huang, who was a student of Randall and then a student of mine, and who initially formulated these enzymes into a silica gel coating. And that was the first time that we could successfully put these enzymes into a coating, and these enzymes would remain active. And this is some of the first experiment that we performed in Duluth Superior Harbor water over the course of 8 weeks. This is the silica gel coating alone. This is the silica gel plus a control biocide, so a molecule that kills microbes and is toxic to other organisms. And this is the silica gel plus the enzyme. And so what we look at here is microbially induced corrosion, because one of the things that some microbes can do also when they grow is to accelerate the process of corrosion because of their metabolisms. There are several ways they can do that. I'm not going to dive into the details of that. But we observed here, as you can, that there is a lot more corrosion in the coating alone than in the coating plus the biocide. And the coating plus the enzyme has the least amount of corrosion. So this was very promising. And this is where we realized that this had some potential to control microbially induced behaviors in the context of aquatic environments. The problem was that these coatings here, this silica-based coatings, they're very weak. They dissolve and they're fairly quickly. And as consequently, the enzyme is just leaking. And while this is a desired trait, for example, for the biocide-containing coatings that are commercially available, because they need to release constantly that amount of toxic ion to constantly kill the surface microbes. In our case, this is not the case. Like, we want to keep the enzyme in the coating because otherwise nothing is going to destroy the communication molecules of the bacteria, and then they're going to take over, right? So we want to keep as much as the enzyme in the coating for as long as we can. So from there, we, with the help of MyCirc, we did significant amount of work in trying to formulate better coatings. So those are just, small examples, and I apologize for the quality of these figures. Somehow they come up blurry here. but we identified ways to reduce leakage. So for example, here, without glutaraldehyde, so glutaraldehyde is a chemical that will covalently immobilize, at least we hope, covalently immobilize the enzyme onto the polymer. So we see without glutaraldehyde, a lot of the activity, enzyme activity, is leaking into the liquid phase, and about 28% of the activity is in the paint. And with glutaraldehyde, more than 90% of the activity remains in the paint, and only about 10% is leaking into the liquid phase. So this is really reducing that leaking issue. And we also verified that these enzymes can be compatible in a variety of different coating base, not just silica coating base, but more commercially available coating base such as acrylic, epoxy, and silicone. And while the activity of the enzyme varies very strongly between the different coatings, you see that, for example, the activity in silicone base is very high for this enzyme compared to the activity in acrylic. There is activity in all of these different coatings that can be detected. One other aspect that is important in this field is to evaluate what is the durability of these coatings. We initially had quite a few issues with this. One thing that's important to consider is that these enzymes, these proteins, are 100% biodegradable. This is a great thing, but we don't necessarily want them to biodegrade too quickly, because otherwise it's a useless product, right? So we want to know that they're going to remain active for a certain amount of time that would make the technology useful in this space. And so this is what we looked at in this experiment. So this is an experiment that was done in the lab, and we are measuring different fraction activity in wet condition in cyan and activity in dry condition in orange. And we're basically seeing that in wet and dry condition, the enzyme activity is remaining fairly constant over the course of 250 days in these new formulation that Colton and Reed in the lab could develop recently. So we're very encouraged by these last results. These coatings with these enzymes were tested also in the field, and this was work that was done with Sichuan. This is an experiment. For example, this was conducted in the Duluth Superior Harbor, this time in acrylic coating. And here you can review some of those sample coupons. This was recently published a few months ago. So you have the no coating control that shows a lot of biocorrosion after 21 months of submersion in your harbor, lots of biocorrosion and lots of fouling. And then you have the acrylic coating control. So this is the coating alone. You have quite a bit after 21 months, quite a bit of corrosion, quite a bit of macrofouling as well, with some mussels there. Then you have the surfactant treatment. So this is a biocide that we used as a positive control. You can see that you have a lot less corrosion, a lot less fouling as well, but you still have significant amount of corrosion, especially after 21 months. And here you have the enzyme acrylic coating, where you have some level of corrosion, but you have less than in the other treatment. And so this is really the longest experiment we've conducted so far, where we see significant activity of this enzymatic coating and significant reduction of the biological process. Here, biocorrosion and biofouling. So you see biofouling was very significantly reduced. This is the same data, just represented differently, measured analytically, where you can see that there is actually statistical significance. Now, how does the enzyme do that exactly? So we know that the enzyme breaks down those signaling molecules, but how is this affecting really corrosion? How is this affecting the attachment of the mussel. Why is that happening? And we don't have the definitive answer to that question by any means, but we have some potential pieces of explanation. What we did with Simon and Randall was to sequence the surface microbiome of these samples. And here you have a big and complex figure, and I'm not going to ask anybody to try to look into all of these little points here. But what you have is a representation of the microbial population on these different samples at 1 month, 2 months, 8 months, 21 months, as a function of treatment here at the bottom. So you have the different treatments. And we see very significant differences for all of these highlighted different taxa here, highlighted by these different arrows. And very interestingly, in this case, so this was done on steel substrate. So in this case, these different taxa here are what we call sulfate-reducing bacteria. So this is a class of bacteria that are reducing sulfate at the surface here, in this case of those steel coupon material. And though this class of bacteria are known to be associated with corrosion. So in this work, we actually showed that the presence of the enzyme changed the community that was present at the surface, and in particular reduced the abundance of this sulfate-reducing bacteria, and that was consistent with the reduction of biocorrosion that was observed there. So this is one way that we think the enzyme alters these biological processes such as biocorrosion and biofouling by altering the surface microbiome population. So in a way, we think that this surface microbiome is in equilibrium. And these bacteria are all coexisting. They're communicating with each other with these different signaling molecules. And all of this is some sort of balance. Now, when we introduce the enzyme here and the enzyme start to chop those signaling molecules, well, that balance is disrupted. And that equilibrium changes, and this change in equilibrium changes the course of event. And the biological processes that are normally very robust and will occur very reproducibly now are on a different course, and we see some changes there. That also means that for us, there is the potential to modify these enzymes so that we potentially target specifically the few bacteria that are most important into the process we're interested in. For example, in regards to zebra mussel, if we know what bacteria are most important to zebras, then we can make potentially enzymes that will specifically target those and minimize the effect on other microbes. And this is some other aspects of these projects here, where this is a little bit of a complicated slide, but it's just to highlight that we can modify these enzymes and their properties so that they will prefer to degrade some signaling molecules as opposed to others. So this would allow us to specifically target some microbes and not others. Now, looking at fouling in more details, shifting now to some of the work of Sichuan and Nathan, where they've done field experiments in Minnetonka and on the Mississippi River. This is some experimental setup that they've used, some cages with suspended coupons here of different material. This is probably polycarbonate here that was coated. During these experiments, we can look at a bunch of different parameters that were similar to those examined in the Duluth experiment. This was an experiment that was performed, I believe, in the Tonka Bay Marina at Lake Minnetonka. Different treatments. This is a protein control. This is the copper oxide. So this is the biocide control. Our two enzymes here. And here we have different indicators that are going to stain different parts of the biofilm. So we have intact cells here in green. We have in red polysaccharide. This is one component of the biofilm, and chlorophyll. This is, for example, some algae. And overall, what you can say is that as you go towards the enzyme, there is less and less intensity in those pictures. So the enzyme, as we thought, is reducing the amount of material, and in particular bacteria, at the surface of the coated surface. Now, this is visible even without confocal microscopy. So those samplings were done after 1 and 2 months in the different sites by Sichuan. We can see here on steel, protein control, biocide control, and our enzyme. We can see a reduction of biocorrosion. We can see a reduction of biofouling here on these polycarbonate samples. It's less and less brown. Similar picture on the Mississippi River here, and in the Tonka Bay Marina, we see a similar picture as well, and a reduction of those circled zebra mussels here as well. So we see here with the measurements that we have less zebras that are attached to the treated coupons. Now, we repeated these experiments later with the help of Reed that measured those samples for 9 months, but also up to 24 months. And he could see some reduction of the biomass and zebras for our lactonase after 9 months of submersion. But what he didn't really see is a strong signal for reduction of biomass and zebra mussel attachment after 14 months, and as well as after 2 years of submersion. And so we think that because those were made with formulations that, some of our first formulations, we think that this might be due to some loss of activity of the enzyme in this coating, and we're excited to be able to test those new improved formulation that we think are a lot more durable in these conditions. Now, very quickly, what's the relationship between surface microbes and zebra mussels? Why do we see that, if we affect surface microbes, we seem to affect zebra mussel attachment? And this is some of the work that Reed has developed. So he developed this system where he places a zebra mussel in a plate and can track photographically its movement. So he just draws a quadrant and measure over time the position of the mussel. And he can do that for a number of these mussels independently and get trajectories for the different animals, for the different mussels. Now, this is what he did. So he did this on pre-established biofilm, and then on plates so biofilm means pre-established community of microbes, and then on plates with no biofilm. And this is the picture he got. So on the pre-established biofilm, the zebra mussels, so here you have 12 different individuals. They seem to move quite a lot. And when they don't have any biofilm on the plate, the movement is largely reduced. So we seem to see that somehow, for one reason that we still don't know, the surface microbiome stimulate zebra mussel movement. Now, the last piece here is that, interestingly, there is also an effect of surface microbiomes and zebra mussel attachment. So he did a similar experiment where this time he has let biofilm grow over time and measured the strength of the zebra mussel attachment. And there is a trend that the more biofilm on the plate and the stronger the attachment of the zebra is. And this is verified in this different experiment here, where he measures the number of zebra mussel that basically remain attached after you invert the plate, and which is another measure of attachment strength. And you can see that indeed, the more biofilm you have, and the stronger the attachment. So there is clearly a relationship between surface microbiome and zebra mussel behavior. And we are very interested into uncovering this and see if we can design some tailored solution to specifically target the microbes that are most important to zebras to reduce their attachment. So the key takeaways from this is that biofilm is a major vector for aquatic invasive species. The current mitigation methods are not great, and some of them are very toxic to the environment. And in this process of biofouling, microbes are extremely important. We have enzymes that can interfere with microbial behaviors, and that these enzymes, they change the surface microbiome, and that change in the surface microbiome, we believe, is responsible for the change in biocorrosion, in biofouling that we observe. And some of the perspectives of that work is that zebra and microbes, they coexist, and somehow they seem to have a very interesting relationship where they influence probably each other's. And we are excited to try to understand how that works so that we can design some very potent and effective and specific, with very limited side effects, solutions to reduce zebra attachment. So overall, we are excited with the potential of these enzymes to be used as eco-friendly additives to coatings to reduce the spread of these invasive species. Here is the group that has performed this work. This is not this year, but this was last year at the State Fair. I'm very grateful to MyCirc for supporting us in doing this work, and also some of our other funding. And thank you very much for your attention today. I'm ready to take any questions.
We'll start with this one. I think this is it's a good one to start with, and I think it'll allow you to expand on something that you just sort of were talking about. This mechanism that links these biofilms to zebra mussel movements and, by extension, zebra mussel attachment. You know, if you were given a stage to just sort of speculate on that mechanism further, what would you hypothesize is going on there? And is it a direct thing? Is it an indirect thing? Is it both? Is it just that these microbes are sort of re-engineering the macro environment that zebra mussels then encounter? Or do you think there's actually some direct relationship there, like maybe they're farming the bacteria or something, or, you know, communicating with them in some other way? What would you guess are the mechanisms there?
Yeah, this is a great question for speculation, but I think there are two big possible answers to this. Like the first potential answer is the zebra mussels and the mussels in general, the shells in general, they only need some sort of mechanical sticky goo to attach. And any mechanical sticky goo you would have made by any bacteria would actually work very well. And this is some of the things that we want to test. Like we want to evaluate whether it's actually specific goo or any goo would work. And then the other explanation is no, it's not just any goo. They are very specific microbes that produce very specific factors that are recruiting the zebra mussels preferentially on site. And of course, the third one is that it's a combination of both. I would say it's equally probable that that one is the major explanation. I know, for example, of a nearly fully resolved case by a colleague at the University of Hawaii, Mike Hatfield, who identified the specific bacteria that is responsible for the recruitment on the surface of a specific insect larva. And it's a specific bacteria, and he even identified the specific chemical that is responsible for its recruitment. So I know that these kind of mechanisms exist, but I can only speculate as to what is happening with the zebras.
I think a very classic question, which is, well, what about the natives? So someone was wondering, is native muscle attachment also inhibited by your coatings, or is there some level of specificity here? In other words, maybe is that attachment mechanism or series of mechanisms specific or more general?
Yeah, it's interesting. It's an interesting question. I don't have a clear answer because frankly, it's because of our experimental design. Like we've really focused our work on zebras, and therefore we've chosen sites that are infested by zebras, and therefore we are mostly seeing zebras. And so I don't really have an answer at this point. I would imagine that it's not necessarily a specific mechanism at this point because we have done nothing to make it specific. So if it was specific, it would be a coincidence. And I don't think it's necessarily an issue because we, for now, envision this solution to be used, for example, on boats. And the adhesion of native mussels on boat is not really something necessarily desirable. So it depends on the final use, but if we envision this kind of use, it might not be an issue. But very interesting question, and would like to test that in the future.
Okay, moving on to the next question here, and maybe there'll be something of a temptation to make this question be more technical. So maybe just thinking about it at a higher level, someone was wondering why sulfate reducers in particular, and why do you think sulfate reducers being affected is having such an impact? Do you think that's just changing the bacterial community and then that's changing something else? Or do you think there actually is maybe a shorter through line there somehow?
Yeah, and this is all really interesting, and I just feel so bad that I don't have more specific answers. But a lot of those sulfate reducing bacteria are bacteria that are using this type of chemical signaling. So some of them are directly affected by the presence of the enzyme because their chemical signaling is being disrupted. And that seems to be a fairly logical explanation as to why their abundance is affected and in that case reduced. However, we know it's not, this is a satisfactory explanation. We do know, however, that there are some other bacterial groups that also use or are known to use this chemical signaling that are, not affected the same way. So there are other mechanisms that we don't know. That bottom line is this is logical, but it's not the only thing that's going on. Now, why sulfate reducing bacteria have an effect on biocorrosion? It's a little bit of a complex mechanism, but to simplify, their metabolism involves oxidation and reduction, and this movement of electrons at the surface of the steel eventually results in accelerated oxidation of the steel and corrosion. There are different mechanisms in other bacteria that directly oxidize iron, etc. But basically, their metabolism alone is sufficient to increase the oxidation of steel.
Super interesting. I'll insert a personal curiosity here for a second, not to get us hung up on what might be a detail, but when you were noting earlier the durability of the enzyme against various solvents, you did note sort of in passing that one of the solvents seemed to elevate the activity. And
Yeah.
do you think that was sort of a fluke or a coincidence, or do you think there was something to that?
No, this is something that we have seen with other denaturing agents. So denaturing agents are are a term that is used in chemistry, in biochemistry, because these molecules typically harm biological molecules. So anything, for example, like a detergent, is a denaturing agent. So the soap we use to wash our hands is a denaturing agent because it basically destroys the structure of most biological molecules. So we've seen in other projects, we've seen that moderate amount of these denaturing agents tend to stimulate the activity of these enzymes, and we think that it increases their flexibility. So in particular, we solve also the crystal structure of these enzymes. So we look, one of my core expertise is actually structural biology. So we look at the atomic structure of these enzymes, and they have one active site loop that binds the substrate, that binds this chemical signaling molecule for degradation. And that loop flexibility, we know, is very important for its activity level. So we think that the denaturing agent increases the flexibility of that loop. And to a certain extent, when that flexibility is increased, it boosts the activity up to a point, of course, where it will be too much and the enzyme will crash.
It's very interesting. We have time for one more here, and I'll grab this one here. It's more of a comment than a question, but sort of a follow-up to that question about native mussels earlier. Someone was noting here that except in very early juvenile stages, native mussels do not have the byssal attachment points. So
Yes.
I'm guessing the implication there being that if what this coating is doing is impacting byssal attachment, then it is less likely to have an impact on native mussels. But you could expand on that a little bit if you can.
Yes, that is a great comment, a great observation, and it's actually a great idea for us to try to understand what is the mechanism, specific mechanism by which these change to the surface community alter the adhesion of these larger macroorganisms. So if we were to look at what is the fate of the adhesion of different macroorganisms that attach differently, then we may be able to understand better how that works. So that's actually a great suggestion for us to try.
Fantastic. I like it. I would like it if this was a two-way street and we gained a lot from each other as a result of this. I'd like to think that we do. All right, I'm going to pause us there or end us there, although I'm sure Mikael would be happy to take any additional questions via
For
email if
sure.
you have some.